Talked all about those different hormonal mechanisms of controlling both blood volume, blood tonicity. Um, and yeah, so these last few slides on that will kind of help us to put all that together and how those things kind of interact with each other. So um, we'll start with a stimulus or kind of like a homeostasis disruption here. And so in this case, the homeostasis disruption is an increase in tonicity. Okay. And so this could be due to fluid loss, right? So if we have fluid loss, great example would be exercise, right? Or just lots of sweating, heat, a hot, humid environment. We lose more fluid than we do salt in that case. And so we can end up with an increase in tonicity, kind of just like an increase in the concentration of our blood due to that, right? Um, and so the stimuli that we're going to be detecting there is obviously going to be high tonicity. Okay, and this is going to be detected by those osmoreceptors in the hypothalamus. Okay, so that's kind of one. That's going to lead to the ADH release that we're going to see. Okay, but then this is also going to be detected by those macula densa cells in that juxtaglomerular apparatus, right? So that's going to sense the high sodium concentration of the filtrate, right? And that is going to be related to the renin release. Okay, so we're going to see a decrease in renin release because of this. Okay, so these are kind of our two stimuli, right? So osmoreceptors in the hypothalamus lead to ADH release, macula densa cells in the juxtaglomerular apparatus, kind of sensing that high sodium, and we're going to see a decrease in renin release. Okay, so with our ADH release, so we're going to see an increase in ADH release, right? And we know that ADH is going to cause mainly the primary thing is an increase in water retention. Primarily in those cells in the collecting duct. Okay, CD for collecting duct. Okay, um, and that's going to help us to dilute our body fluids. All right, so we're holding on to more water as fluid is passing through the kidneys. Okay, um, and then we see this decrease in renin release. Okay, this is because we've already got plenty of sodium, right? We got plenty of sodium, so we're going to decrease renin release here, actually. And so this is going to cause in the end, a decrease in sodium retention, right? And a lot of that has to do with decrease in aldosterone release. Okay, so decreased aldosterone release um, can be caused by a decrease in angiotensin 2 that we would see from that decrease in renin, right? Um, and so those are kind of the key mechanisms there. And the way we want to think about this when we're trying to put things together with ADH and renin and aldosterone and angiotensin 2 is that the renin part is really focused on sodium levels in the body, and the ADH or vasopressin release is going to be focused on water in general, right? As far as like its effects, okay? And so, yeah, so then our last piece here is our GFR is going to change, right? So if we have high sodium levels, like we're talking about, we're going to see a decrease in, we're going to see a constriction of the afferent arteriole. And we're going to see contraction of those mesangial cells. Oops. All right, contraction, put it right here. Okay, so all of this together is going to decrease our GFR. right? Because we're trying not to lose water, right? We already don't have enough water, right? We got too concentrated of fluid, right? Not enough water, too much salt. So we're trying not to lose as much water. So we're going to decrease GFR. Okay. All right. So, yeah, I know. You okay? Yeah. Too much water. Just kidding. Not enough water, maybe. I don't know. Okay. All right. Yeah, so that's kind of our first one. The next one we want to talk about is when we have a disruption in volume. Okay. Um, and so yeah, so with this one, I want you guys to take a stab at this first. Okay, we'll start out just kind of giving you an intro to it. So hormonal control of volume, right? So when blood volume decreases, okay, due to fluid loss, what happens is, and so this could be, yeah, this could be bleeding, this could be just loss of volume when we have kind of an equal amount of water and salt loss, okay? Um, and so we're going to be thinking about both baroreceptors here. Okay, that can be those granular cells. Okay, and then we also have low pressure receptors. And so those will be like in the atria in those places like we talked about, okay? So those are both going to be, so we can think about this first one being connected to renin release. We can think about this one being connected to ADH release, okay? So go ahead and just work through that a little bit. Decide what you think each of these are going to cause, and then what do we see for ADH release? What do you see for renin release? What do you think our GFR needs to do in this case? Okay, so go ahead and work through that. We'll come back together in just a couple minutes. Yeah.
When you use the word retention versus
Yeah.
absorption, retention means like relative to the blood plasma.
Yeah.
The actual
Retention is just a little bit more general, kind of like we're holding on to more of that somehow, right? So it could be that we're making less filtrate and then we wouldn't have to reabsorb it, right? Or it could be that we made filtrate and then we reabsorbed it. So I'm using retention to kind of like capture both those ideas.
Would it be accurate to say EDH helps retain more water? Or would it is more accurate to say it absorbs more?
Both would be accurate for sure. Yeah. Yeah.
Cool, thank you.
Yeah.
Do ADH or renin, or I guess renin specifically, does it increase the reabsorption and decrease the output of sodium? Or is the sodium always going to go
Renin
out?
yourself, you're saying? Or
Yeah, just
renin
renin,
itself?
yeah.
Yeah, so renin, by its, yeah, let's go back to by its effects on aldosterone, for sure, will then cause more reabsorption of sodium. But you're asking, does it have that action by itself? Renin?
Yeah.
Yeah, renin doesn't really have actions by itself. It's really just all about causing the formation of angiotensin 2.
Okay.
And then aldosterone release are the big ones. But that's what I wanted to check and just make sure with the actions of angiotensin 2, there's so many. Does it directly cause sodium reabsorption? I think it does. Where is that one here? Increased thirst and salt. Yeah, that last bullet point there, direct effect on kidney tubule cells to increase sodium reabsorption.
And
So
the
yeah.
contraction of the mesenterial cells makes it less permeable to sodium as well, even though it's such a small molecule?
Sure, it's just about those things going together, right?
Okay.
When we create the filtrators, water and sodium go together. So it's just less of that
Okay.
in total. Yeah.
Yeah.
Mhm. Yeah,
Thank you.
cool, yeah.
I don't make this. It takes a long time, but I think it helps.
We're talking about best studies.
We are.
Talking about what?
Best study method.
Oh, best study methods, okay. What are you? I'd love to hear them.
Flashcards, supports.
Yeah,
I know.
writing stuff out, yeah.
Yeah.
Those are the two. Those are the two things I always recommend. Yeah. Yeah.
Thus far.
Remaking them, yeah, like the process of doing this, yeah.
The slides.
Maybe I'll adopt that. And I've done different things, but she's made Aussie, so I tried those. I'm like, I've seen him on.
I was gonna say, and I drive by her and she's listening to my stuff and honk at her, yeah.
You're like.
I know, I took like them into Notebook LM and.
Yeah, make a podcast, that's awesome, yeah, those are helpful.
I just
Yeah.
Yeah.
Yeah, and why don't you guys go ahead and just move into the next one too, because it's just the opposite. But it's a little bit different because it'll add in the A and P part of it as well. But it's just when we have too much volume, right? So why don't you go ahead and move into that one too?
Yeah.
Type of volume. Yeah. So, yeah, well, the ones for the H is the ones for the.
But like, stuff funny, you don't know everything about.
And then I go back and
I was it. I said, you didn't respond. I was like, I'm just looking at it.
And for the most part, all of them. And then for
Oh, you, yeah.
All right, guys, let's go ahead and come back together here. Okay, so let's start with this first one, which would be a blood volume decrease, right? So if our main stimulus is low blood volume, okay, we got to think about the two primary places that stimulus, or the stimulus would be being detected. So the baroreceptors, which are the granular cells in our kidneys, okay, in our juxtaglomerular apparatus and that afferent arteriole, right? So those are going to sense Low blood flow, right? Like we talked about as being the stimulus there. So that low blood flow is then going to lead to the release of renin, right? So we're going to increase renin release. Okay. These low pressure receptors, mostly in the atria, but other parts around the heart as well, are going to sense again low pressure. All right, and that low pressure is going to lead to an increase in ADH release. Okay, so that's kind of our stimuli. And we could kind of go into all the details of this one, and you should be able to go into all the details of that one, right? So increase in renin. Again, renin is really just an enzyme, right? And so it's going to turn angiotensinogen into angiotensin 1. Angiotensin 1 is going to get turned into angiotensin 2 by the ACE enzyme in the lungs mostly. And then we got angiotensin 2, right? So we can kind of, since I already started down that path, we can kind of go here first. So overall, we're going to get an increase in angiotensin 2. Okay. We're going to get an increase in aldosterone. Okay. And that's really caused by the angiotensin 2, right? It's probably easier just to kind of just take a peek back on the angiotensin 2 slide and instead of me trying to cram all that in there, because there's so many actions, right? It's all of these actions of angiotensin 2, right? So let's just kind of review those. So it's a vasoconstrictor, right? So that's really important is we're vasoconstricting. That's its strongest mechanism. Causes us to be thirsty, causes us to want more salt as well. Causes some ADH secretion, okay? Causes ACTH secretion, which is going to lead to aldosterone release, okay? Causes aldosterone secretion directly at the adrenal cortex, right? So that's going to then lead to sodium retention or sodium reabsorption, right? And potassium excretion. It causes us to release more norepinephrine from our sympathetic neurons, which then that's going to lead to some vasoconstriction. And it's going to lead to some renin release, as we talked about previously on the stimuli for renin release, which we can go back to real quick right here, right? Sympathetic activity, norepinephrine on these cells in here, these lacy cells and these granular cells, they're stimulated by beta adrenergic receptors to release renin. Okay, so that's how we're connecting this action here, right, to more renin release. We contract those mesangial cells. That's going to decrease our capillary permeability, decrease our GFR, right? Makes sense. Make less filtrate, right? Urinate less because we don't have enough fluid. And then we also directly cause more sodium reabsorption as well, right? Which is going to also pull more water back too, right? So water's going to follow that sodium. Okay, so lots of things going on there with angiotensin 2. So when you think about renin being released, that's what you're thinking about, right? Is all the effects of angiotensin 2, right? So yeah, you got to make sure you make that connection, right? Renin really translates to angiotensin 2. And then when angiotensin 2 is released, we got that whole host of effects, right? It's the most powerful mechanism for both salt and water retention. But again, this one's kind of primarily driven by primarily helping us with the salt part of things. And water follows that too, right? But sodium is kind of the primary concern of the whole renin system, whereas water is kind of the primary concern of vasopressin or ADH. Okay. So what should I actually write there? I'm not sure. But you could really just write all of those things that we have on the actions of angiotensin 2, which is this slide, right? So all of these things, okay? But to summarize it, we'll just say increase in sodium reabsorption. Okay, we're going to decrease GFR because of this, right? We're going to cause some thirst. Going to cause some salt craving. Okay. This is going to connect to more ADH release as well, but the big ones here are these guys, right? And of course, water is going to follow that sodium. Okay. Then we got ADH release, right? So ADH release, we're going to increase ADH release here as well. And again, we're going to increase water reabsorption. Most of that's at the collecting duct. Okay, we can also go back to that ADH or vasopressin slide where we had all of the actions, right? Because we've got this is the big one, right? So this is bringing back the water, okay? But it's also a vasoconstrictor too, right? So vasoconstriction is a part of that as well. And it causes some ACTH release, which is then going to cause some aldosterone release. That's a minor effect. But the big ones are water reabsorption as well as some vasoconstriction. Right. And again, we've got kind of really three ways, but kind of two ways to increase blood pressure, right? One is make the pipe smaller. Two is get more water in those pipes somehow, right? So we get more water in them by drinking more, right? Or we can get more water in them by not losing as much water, right? And then we can make the pipe smaller by vasoconstriction, right? So we're kind of utilizing all of those mechanisms by keeping more water that we did drink, right? Causing us to drink more water, and making the pipe smaller with these things here. Okay. So this is kind of the basic effects, right? But I wanted to point back to those other slides too, because we see all of the effects of those hormones too that you definitely want to be able to spell out in these scenarios. Okay, so GFR is going to go down, right? And how does it go down? It's going to go down by vasoconstriction of the afferent arteriole. Okay, there's two things that cause that we talked about, right? One of those things is norepinephrine. The other one is angiotensin 2. Okay, so that's just decreasing the amount of hydrostatic pressure in the glomerulus, right? We're decreasing the amount of hydrostatic pressure by sending less blood into the glomerulus, right? So slowing down the flow to it, okay? So that's one, again, you want to think about it in bigger terms of like, how do we change the amount of filtrate we produce, right? One would be decrease hydrostatic pressure into the glomerulus, right? Two would be make the glomerulus less permeable too, right? And that's also what we do, right? We also, I'm just going to put it right here, contract those mesangial cells. Okay, that's an effect of angiotensin 2 as well. Okay, so we make the glomerulus less permeable, right? And we send less blood in there, so we decrease hydrostatic pressure. So we're going to decrease our GFR with those two primary ways. Okay, what questions do we have on that? Yeah, go ahead, Kyle.
Does aldosterone also contribute to the afferent?
I believe it does. Let's go back. I do mix all these effects up, so I want to make sure I'm telling you the right thing. There it is. Yep. Last bullet point there. Thank you. Yep. Constricts the afferent arteriole as well. Thank you. Yeah. Good question. Good point. Other questions? Yeah.
So when you say contract mesangial cells,
Yeah.
that's pointing to angiotensin 2.
Yeah.
Just a result of it.
Causing that. Yeah. So it should be the arrow should kind of be the other way, right? Angiotensin 2 is causing the contraction of those mesangial cells. Yeah. Yeah. Okay. What else? Other questions on that? Okay. All right. Well, then let's look at the other way with when our fluid volume increases, right? We got too much volume here, right? So again, we've got our granular cells, right? Are going to result in a decrease in the release of renin, right? And now we've got high pressure receptors in the large veins, or sorry, high pressure in those large veins. Okay, as well as the atria. And that's going to lead to a decrease in ADH. All right, but now we've got a third mechanism here, which would be stretch of the atria, which is going to lead to increased ANP as well as BNP. BNP was just first discovered in the brain. That's why it had initially had that B name, but it's also called B-type natriuretic peptide. But they have similar effects, ANP and BNP, both released by the heart, by the atria, and the ventricles as well. Okay, so we got ADH, we have a decrease in ADH release, so we're going to reabsorb less water. All right, we're going to decrease vasoconstriction. Okay, we're going to reverse all the stuff we just talked about with angiotensin 2 and aldosterone, right? So in general, we're going to lose more sodium. All right, we're going to constrict less. All right, we're going to decrease thirst. All of those things we just talked about, just kind of the reverse of those things. So we're going to let go of sodium, going to constrict our blood vessels less. All right, we're gonna decrease blood pressure instead of increase blood pressure here. Okay, we're gonna increase GFR. All right, and that's going to be, yes, we're going to increase it there. All right, so those mechanisms are dilate the afferent arteriole. We're going to relax mesangial cells. All right, so we're going to decrease hydrostatic pressure. We're going to increase permeability. Okay, so ANP, along with the two mechanisms we talked about on the last slide with angiotensin 2 and aldosterone and norepinephrine, those are all reversing, right, for the afferent arteriole and for the mesangial cells. Along with that, ANP does both of these things as well. ANP contributes to both of these factors here, dilating the afferent arteriole and relaxing the mesangial cells. Yeah, the easiest way to kind of summarize what AMP is doing is it's just pretty much the reverse of the previous slide. All right, and so we can look back on this slide right here to look at the actions, right? It's going to block ADH release, decrease renin release, decrease aldosterone release, dilation of the afferent arterioles, and that relaxation of the mesangial cells, which I don't even know if I have written on there, but that is another effect of it. Maybe I have it on this slide. Yeah, I have it on that slide there. Yeah. Okay, so kind of just think about it as doing the exact opposite of everything we were just talking about on the previous slide. It's our form of shedding as much water and salt as possible, right? Which is not normally the state that we're in, right? We're normally trying to preserve all of that stuff. Yeah, go ahead. Say that one more time. You're asking what I wrote there? Yeah, decrease thirst and increase GFR. Yeah. I was so surprised that you couldn't read my writing. I didn't know what your question was. I'm just kidding. Yeah. Okay.
There's slide where we have bus into the sodium. Would
This one here?
The blood tonicity increases the valves. Would the effect of renin be lower because because while we are getting rid of the sodium, we're also kind of getting rid of water along with it. And we don't want to do that if we have loss.
Yeah, so you're kind of, because this also leads to
Increase thirst.
yeah, all those things too, for sure. Yeah, so we don't have as powerful of a stimulus to, yeah, be thirsty, be all those other things, to vasoconstrict, all those other things, right? We kind of have half of the mechanism for increasing blood pressure here. And so, yeah, so it's mostly going to be, yeah, it's just not as powerful of an effect, but it's kind of trying to balance out that just that out of whack distribution of salt and water, right? By mostly just pulling the water back and not focusing so much on the sodium, which will happen with aldosterone and angiotensin II. And so yeah, because of that, we don't get as powerful a vasoconstriction. We don't get as drastic of an increase in so in body fluid retention because we're not utilizing sodium as much to do that, right? We're mainly just focused on the water part of it, right? And so yeah, it's not as powerful of a fluid retention mechanism as when we put it all into play here, right? This is kind of all hands on deck, using everything we can to pull that water back. But if tonicity is messed up, then that's a priority as well. So that needs to get fixed, which is why we're kind of seeing this juxtaposition here of these two different things. So yeah, good question. Okay, other questions? Yeah, Kyle.
On the very last side for the increase in volume, if ADH is down, we also say that that will contribute to a decrease in ACTH release. But those there's like less in line over here than median.
Yeah, so ADH goes down because of the decrease in angiotensin II. We'll have less ACTH release, which then we would have less aldosterone release. Is that answering your question or not quite?
V1A and then V1B receptors.
Oh, I see. Yeah. I see what you're referring to, the last effect of vasopressin, which was here. Yeah, this part right here, right? Yeah, so we get a little bit less of that as well, which then leads to less aldosterone secretion as well. Yeah, so part of the mechanism for that. I believe this isn't as powerful as the angiotensin II mechanism. So the loss of angiotensin II will probably have a greater effect on the decrease in aldosterone. But yeah, this contributes as well. Yeah, great question. Was there another question over here? Okay. All right. Well, let's take a short break before we get into the pH balance stuff, which will be next. Okay. Let's just take a 5-minute bathroom break. Come back right after 10:10, and then we'll finish this up. All right. Let's go ahead and dive into this last part of this, which is the pH balance and how the kidneys help us out with that. We talked about this even on the very first day of class, talking about metabolic acidosis and things like that, and kind of how the kidneys can help us. So we've seen these ideas for quite a while, right? Kidneys can help us to get rid of hydrogen or bicarbonate or the reverse. Okay. And the main way it's done is through that same enzyme we talked about when we talked about the chloride shift, right? That carbonic anhydrase. We have it both inside our kidney tubule cells and then also on the membranes of our kidney tubule cells into the lumen of the nephron. Okay. And so these I cells are the ones we're talking about now that help us do that. Okay. And so we're usually trying to get rid of hydrogen. That's the normal state of our body, right? Is we're normally moving towards a lower pH. Right. And so we're trying to get rid of hydrogen and conserve bicarbonate, but it's not always the case, right? But so that's the mechanism we'll walk through is what it looks like when we're trying to raise our pH back up. Urine's usually acidic because we're getting rid of that hydrogen, right? But yes, it can go either way. So let's just walk through this mechanism here. Okay, so we've got the blood over here. So this would be kind of interstitial space here, right? This is the tubular fluid, right? So that's where the filtrate is being, yeah, that's where the filtrate is, okay? And so the mechanism for secreting hydrogen ions and conserving or reabsorbing bicarbonate actually starts with filtered hydrogen and bicarbonate. Okay, we can see those guys right here. Okay, so those came from the glomerulus, right? When we made the filtrate, because they just went with the water, right? When we made that filtrate. So they're now in the filtrate, okay? And so we're going to then have them combine with the help of carbonic anhydrase here. We can see that here, right? First of all, we're going to combine into that CO. First of all, make carbonic acid, H2CO3, and then that's going to dissociate into CO2 and water, right? This is where the carbonic anhydrase helps right there, as we can see that happening right there, okay? And then the CO2 goes into the tubule cell, the kidney tubule cell. All right, so we can see that CO2 going into the kidney tubule cell. Okay, the water can stay there, or it can follow another ion, depending on where we're at in the nephron. Okay, so the kidney tubule cell pretty much reabsorbs that CO2. Okay, and now we go back through that same process. We combine CO2 and water. Right, that's going to be with the help of carbonic anhydrase again. All right, we're going to go back to carbonic acid, and then we're going to go back to making this hydrogen and bicarbonate. Okay, so we just do that same, we do that process in the lumen of the in the filtrate, and then we take the CO2 in, do that same process again inside the kidney tubule cell. Okay, so now we can see we can reabsorb some of that water here, right? We can take that back in. We can then send out the hydrogen. So hydrogen secreted via two primary mechanisms. So it could be just primary active transport. All right, that one's shown here. Right, we see the ATP being used to just pump hydrogen into the filtrate. All right, so that'd be primary active transport, or we can do that in exchange for sodium. All right, we see that one right here. Sodium proton exchanger. Okay, and so aldosterone actually activates more of this happening, right? So it leads to the formation of more sodium channels, which makes sense, but it also leads to the formation of more of these sodium hydrogen exchangers too, right? So it's going to help us to pull more sodium back, but also get rid of more hydrogen at the same time, okay? And then, yeah, so so this one is happening mostly at the proximal tubule. And then these pumps are more common in the distal convoluted tubule. This is supposed to be a D there. Sorry about that. Distal convoluted tubule, proximal convoluted tubule. We can see both mechanisms in either place, but it's more common to have those as I wrote it there. Okay. Yeah, and then the last thing is bicarbonate is moved back into the blood via chloride exchange. Right, we see that mechanism right there. So that's the same type of transporter we see on the red blood cell, right? That's going to do that for us in the chloride exchange. Okay, so this doesn't have to just be CO2 that was formed this way. We can also end up with CO2 in the filtrate just from the byproduct of metabolism too, right? We'll have CO2 floating around in our plasma in that same way. Okay, questions on those steps? Yeah, go ahead.
The bicarbonate and chlorine exchange, is that the same as what would happen in hemoglobin? Is it for the same reasoning? Or
Yeah, Pretty much the same reason, yeah. Uh-huh. Yeah. And then the only other mechanism I didn't mention is we can also move bicarbonate and co-transport with sodium there as well. It's just a different mechanism, but that helps us in both ways too, right? Getting the sodium back and the bicarbonate back that way. Yeah, Priscilla. I mean Tiff, sorry. I said the wrong name. Yeah. What's that?
Is it a? Oh, sorry.
Which one are you talking about? Where are you talking about over here where I wrote stuff? Yeah. Ca. They're also the two places I circled things were Ca. Yeah. So carbonic anhydrase. Yeah. Yeah. Okay. Other questions on that? One thing that's just interesting that I found that I added here, just to remind me to talk about it, is my family and I did a big trip when my wife and I were both on sabbatical, and a month of that we spent in Quito, Ecuador, and Quito's at like 10,000 feet elevation, and so a lot of people when you're traveling there, you can be prescribed acetazolamide to help you adjust with the altitude. Has anybody taken that for altitude? Yeah, it was kind of it's a lot of people get altitude sickness just going to Quito because you just fly in and you're there all of a sudden at 10,000 feet instead of any kind of acclimation process. And I was just super curious to how it works. But it's actually a carbonic anhydrase antagonist. That's what acetazolamide is. So pretty much what it does is it leads to more bicarbonate excretion, and we lose more bicarbonate. And because of that, it results in a little bit of metabolic acidosis, which then makes us breathe a little bit more, which then helps us kind of saturate our blood a little bit more through that process we talked about before with that hyperventilation that we get at altitude. So it kind of kicks off that process early instead of waiting for the hypoxemia elicited by the altitude to do that. It kind of forces that process early, which yeah, I just thought was interesting. Don't be too worried about that if you didn't quite follow all that out. Just kind of extra information. But yeah, just
So you're saying that at the proximal convoluted tubule.
Yeah.
There's sodium exchange and then the distal convoluted tubule is primary active transport.
yeah, and both can happen at either place, but it's kind of this is the primary mechanism at the proximal convoluted tubule. This is the primary mechanism at the distal convoluted tubule. Yeah. Yeah. Okay. Other questions on that? All right. So what happens then afterwards? Okay, so we're pushing that hydrogen back into the urine. Okay. Well, those hydrogen pumps in the kidneys we were just talking about, they max out at a pH of about 4.5, which is not that strongly acidic for our urine. Okay. So this would be reached quickly if we didn't have the hydrogen ions bind to something in the urine. Okay. So we need to have some buffering that happens in the urine so we don't build up hydrogen ions too much. So then we can't push any more into the urine. Right. So there's a few things that can help us with that. Number one is the bicarbonate that's there. Okay, so there's some bicarbonate in the urine that can buffer it, right? There is phosphate buffer in the urine as well. Okay, and then there is ammonia. Okay, and so we'll talk about the ammonia mechanism in a second, but this one is kind of a 2-for-1 deal because we need to get rid of nitrogen as well, and so those kind of help each other out. We'll talk about that, how that works in just a sec. Okay. All right, so just a quick note on why do we have all this extra hydrogen ions that we need to get rid of, right? So one of the main things is certain amino acids, when used to produce glucose and gluconeogenesis, produce very strong acids that make a lot of hydrogen ions, okay? So that's kind of showing this process right here, okay? CO2 from metabolism is forces our pH down as well. Most of our CO2 is breathed out of our lungs, so it's not as big of we don't need to rely on our kidneys as much for this, but it is a contributor to this. Obviously, things like exercise can add to the hydrogen ion load in our body, any abnormal conditions like ketosis, and then just ingesting foods and drinks that are acidic as well. So all of these things contribute to this idea that we need to be constantly getting rid of hydrogen ions in our urine. Okay, so then connecting this ammonia secretion to the hydrogen ion secretion. Okay, so two things in our body that we're constantly breaking down and replenishing are proteins and nucleic acids, right? These are the two nitrogenous compounds that are really important organic molecules in our body, but they contain nitrogen, right? So when we break these down and kind of recycle those, we need to deal with that nitrogenous waste. Okay, and so that produces ammonium. Okay, so ammonium is NH4. Okay, that's a byproduct of breaking down these proteins and nucleic acids. Okay, and so we'll kind of go through the process, just the steps here in the kidneys of how we get rid of that ammonium. Okay, so out here, we're going to start out here in the paratubular space, which would just be like the interstitial space, right? So same orientation as we were before. This is the filtrate of the urine. This is the kidney tubule cell. This is the interstitial space, right? So from the blood, we're sending ammonium into this interstitial space, which then we're going to try to get into the kidney cells. But we can't get NH4 across cell membranes. Okay, so ammonium doesn't get across cell membranes. So we need to convert it into NH3 first. Okay, so NH4 in interstitial space. Okay, we're going to convert into ammonia, NH3. Okay, and then we can get it into those tubule cells. Okay, so we see that happening right here. NH4 converted into NH3, gets into the tubule cells. It's NH3 here, okay, and then we can again move that across the membrane in the form of NH3, and now we got it into the filtrate. Okay, so then NH3 into filtrate. Okay, then our last step to kind of keep it in the urine is to add a hydrogen ion to it, right? So we can use it as a buffer. So then we get that NH3 plus H plus, that turns into NH4, and now it's trapped in the urine, right? So we're trapping the nitrogen in the urine by adding a hydrogen ion to it, and we're using this as a buffer, right? We're using this to absorb some of those hydrogen ions that we added to the urine before. So we keep them there. We keep the pH high enough to where we can continue to pump more hydrogen ions in, and we trap that ammonia and now ammonium in the urine so we can get rid of it. Okay, so this is called ammonia trapping in the urine. Okay, so it's kind of a 2-for-1 deal there. Okay, questions on that at all? All right, let's have you guys do a quick kind of drawing of both those things. Okay, so we got our urine over here, our filtrate, we got our kidney tubule cell, we got our blood. Let's have you draw the steps of hydrogen ion secretion that we went through, as well as bicarbonate reabsorption, as well as the ammonia trapping that we just went through, kind of combine all of that into one drawing. I'll draw it as well, but you guys got to make sure to draw it. Don't wait till you see mine. Okay, so go ahead, work on that with the people around you, and we'll finish up here in a sec.
Oh, yeah, there's two verses. I didn't move this one. Yes. Wait, it's bad. Oh, you know, it's funny. Because that's where I'm saying, look back. What's happening here? One of these.
CO3.
It's not just reading that.
I get
It's it's
You can do it. Ben. No, you said I can't do that? Or
No,
what did you say?
it's like in class, like,
Mm.
I just have a hard time like paying attention to following along.
Totally.
So like right now that I'm doing this, I'm like, wait, let me go back.
Yeah, there you go. That's why I have you stop and do this stuff, because it's so hard to. Yeah.
Not so much.
Yeah, yeah, just loving it, yeah. Some bedtime stories, physiology bedtime stories, put you to sleep.
Hey, look, mine's really hard.
I want to show yours, Margot. I like yours. I want to show yours to the class, like your little color things there. That'd be cool.
You're doing currently.
Oh, like yours, like, yeah.
I'm working hard.
Yeah. Yeah, that's all right. Yeah.
I can't.
Morgan, you can do this.
I keep trying, but it's not. I can't do like the split screens. I was trying to figure it out.
I see. Gotcha, yeah. I see, I see. You don't have it all memorized yet after the first time you heard it? Just kidding. There's a PA student, I think, he's in his last year now, but he didn't write a single note in this class ever. He just sat there and like looked the whole time.
I feel like that was Sophie with your class. She's
What?
taking busy on
Yeah,
how.
it's true. Yeah, I know. But yeah, this is the way he was in undergrad too. He would just like, he would just look at me. And he was just like, yeah, John.
Oh,
I
I
can't think of his last name right now.
think I have a picture.
He's graduating with Sophie's class, but yeah, he would just stare at me, and that's just the way his brain works. Like, yeah, it just doesn't work when I write stuff down. I just have to just listen. Yeah, no, thanks. Dang, yeah.
Maybe volume.
Whoa, that's good. I know. Hello.
Just got it. You're good. Moving on. What's so funny?
Hang on.
Why are you laughing?
Oh, is that gonna be in the recording too?
Yes, it is in the record, yeah. Oh, nice. You know, too, we got multiple, yeah, multiple things to back it up. Okay, looks like we're mostly done. Okay. All right, let's, I'll just show you what I drew here. Probably very similar to what you drew, I'm sure. I didn't do any color coding, I'm sorry. But yeah, let's just start over here, right? So we got filtered bicarbonate and hydrogen, combines, turns into carbonic acid. Through the help of carbonic anhydrase, we break that down into water and CO2, right? The CO2 goes into the kidney tubule cell, again combines with water with the help of carbonic anhydrase, back into carbonic acid, splits into hydrogen and bicarbonate, right? The hydrogen can get pumped out just directly via hydrogen pumps, via ATP, primary active transport, or an exchange with sodium here, okay? The bicarbonate can be put back into the blood in exchange for chloride, or with co-transport with sodium there. Okay, so that's kind of the pH part. And then we've got the ammonium over here, which came from the blood, went into the interstitial space. I didn't really leave myself much interstitial space there. But in the interstitial space, it gets converted into ammonia, which can be transported through channels into the kidney tubule cell. And then again, out of the kidney tubule cell as ammonia, NH3, combines with maybe one of those hydrogen ions we sent in right there to get ammonium. And now it's trapped in the urine. Okay. Questions on any of that? Okay. Give you a sec to finish writing that down. Yeah. No, it occurs in both the proximal convoluted tubule and the distal tubule as well. Yeah. Mm-hmm. Other questions on this? Yeah, go ahead, Dana. Yeah.
Either, no transport with chloride or sodium.
It would be exchange with chloride, go in the opposite direction, right? And then co-transport with sodium, go in the same direction. Yeah. Mm-hmm.
Does the co-transport require
Yeah, good question. I'm not sure. Yeah, I don't know what that mechanism is. Yeah, good question. Other questions? Okay, I still see some of you writing, so I'll just give you a sec. All right. Well, I just wanted to kind of say a couple things to kind of wrap us up here. So let's do that. And then I'll put a QR code up for the course evaluation. And I'm supposed to leave when you guys are doing the course evaluation. That's just best practice. So so just warning you, I'll do that. But okay, so a couple things. Back to week 1 of this class, okay? I was trying to convince you why you should love physiology. Some of you didn't need any convincing at all. Some of you still do, for sure, and that's okay. But yeah, we talked about how it's the why behind so much of what you do as a PA, right? The best clinicians are going to understand the mechanisms for everything that they do, right? The best clinicians can also explain the mechanisms, right? And hopefully explain the mechanisms in a way that makes sense to your patients, right? And one thing I wanted to say is this was hard, right? This class was hard. It still is hard. You still got to test, right? But one of the great things about the way the human body is designed is that we don't grow unless we're challenged, right? And I hope that you're different after this semester, not just because of this class, but maybe hopefully because of this class too. But I hope that you actually are changed because of the challenges you faced, right? And that is how our body adapts to everything, whether it's a physical stress, a mental stress, a educational stress, I guess. That's how we change. That's how we grow. So it's worth the hard in the end. And yeah, so a couple things. Really, my whole foundation of why I love studying physiology and the body so much is because I think it's a window into seeing God and to seeing our Creator. And Romans 1:20, I think, says it best that God's invisible qualities have been clearly seen through by understanding what's been made, right? We can see God, we can know God actually through studying His creation. And I think that's a beautiful thing. So I just have a few things for you in the form of hopes for you as we wrap this up. So number one, I hope you found yourself in awe of this amazing stuff at some point this semester, maybe multiple points this semester. Um, I hope that your understanding of this can actually help others experience that awe or that peek into God as well, right? That's the power that you have as somebody who understands something about the body, is you can share that with somebody else and then go, whoa, that's how it works. That's so cool, right? And that's amazing. I hope that your respect for the human body translates into actually treating people with respect as well, right? And treating people with respect as image bearers of God, right? As we all are. And yeah, so with each patient you encounter, I hope you'll not just rely on routine, experience, and protocol, but you'll consider the physiology and how it's supposed to be working and see how the disease state differs from that. Same thing with drugs or treatments, right? Not just kind of relying on your protocol, but understanding how it's supposed to work, how it's not working in the pathophys, right? And how the drug or treatment changes that pathophys and kind of gets us back more towards normal physiology, right? This one I love. In humility, I hope that you'll look for opportunities to learn every day, right? Because the this psychological principle called the Dunning-Kruger effect is something that I come up against every day with your class, with your questions in class, and just in studying everything. Is like there's always something more to know, right? And the actual secret of all of this is like the people who know the most actually understand that they know what they don't know the most as well, right? And so that's the Dunning-Kruger effect, is that yeah, the more you know, the more acutely you know what you don't know as well, right? And that you just get this world opened up to you. There's so much to know, right? And so hopefully I hope that you'll use your knowledge of how the body works to educate your patients, colleagues in a humble and understanding way. So yeah, just a little bit of my hopes for you. And yeah, I get to come back in the spring and do a couple of days of class on kind of my side passion project, which is obesity and weight stigma. And so we'll dive into that at some point in your medicine and society class, I think, the one that Dr. Miller leads. So I'll see you guys again then, but definitely see you around a lot. And please stay in touch, because this is really my only major touch with you guys in this program. But I love hearing from you and seeing you around the hallways and stuff. It might take me 30 seconds to get your name back when I see you, if I haven't seen you for like a year, but it usually does eventually come back. So yeah, don't be a stranger. Okay. All right, guys. So I'd love if you just filled out the course eval with your honest feedback, for sure. So this should take you there. You should also have an email about it too, maybe. But yeah, so yeah, I'll be seeing you guys next week on Tuesday. Okay, thanks for a great semester, guys. Yeah.